All right, in this video, the goal is to go through a pretty thorough coverage of anatomy and physiology of the eye. So we're mostly going to focus on the actual sensory, like visual sensation part of it, but we'll add in a few other things along the way. So as a quick introduction, we need to talk about what the visual system is even trying to accomplish. Of course, the end goal is to make an image somewhere in your brain in the visual cortex that you can use to see the world around you. And we're going to have to use a handful of somewhat technical terms to make sense of that. The first one, photosensitivity. You can think of this as asking the question, how much light, right? How much light does it take in order to get a reliable image? So in this case, right, we've got two different types of photoreceptor cells in the eye, rods and cones. And the general distinction here is that if you compare them, right, rods versus cones, the rods tend to have a very high sensitivity, right? And cones have relatively low sensitivity. All that means is that the rods are going to be pretty good at night vision when lights are dim. The cones need bright light to make that happen, right? The other factor that we care about, though, is resolution. Think of resolution as like pixels, right? When you say that a camera has 10 megapixels, that's just how many little dots make up the image. The downside of rods is they have low resolution. The image looks kind of grainy. That's why you don't see very well in dim light. Whereas cones have high resolution, right? So assuming the lights are bright, we have every reason to use cones. They give us better imaging. You can see everything more clearly. And the only reason that you would use the rods is if the lights are dim enough that the cones don't really work anymore. Okay, so that's sensitivity and resolution. The relaying the signal to the brain, right? Once we get a signal in the rods and cones of the retina, we have to tell the brain what's going on. That is going to be cranial nerve 2, the optic nerve, right? That is the nerve coming off the back of the retina. Really, really large nerve, taking tons of information because you have, millions of photoreceptors, right? So a lot of data going back. All right, focus. Focus is entirely a product of your lens, and it's asking the question, how far away is the object that you're seeing, right? So focus is like a near versus far thing. Just like with a digital camera, if you're out of focus, things that you're imaging in different planes, like how far away they are from the camera, are going to look blurry, right? If you focus on someone very close to you, the people in the background look blurry. If you focus at the back of the room, the people in the front look blurry. It's impossible to make them all look sharp. Your lens in the eye is accomplishing that same thing, right? And then lastly, color discrimination. We don't want to just see in black and white. And so this is entirely dependent on cones. You actually have 3 different types of cones that compare different wavelengths or colors of light, and we'll get to that before too long. All right, so a quick word on some basic anatomy. When we're tracing light through the eye, the main thing that I want to hit here is obviously light has to come in from the outside world, and the first structure it's going to go through is the cornea. So light hits your cornea, and since the cornea is curved, that is going to bend light through an opening that we call the pupil, right? So the iris is the colored tissue. So when you say you have brown eyes or blue eyes, the colored portion is the iris. The opening, the black circle in the middle, is the pupil. We're directing light through the pupil. And then the next thing it hits is the lens. Light also bends when it hits the lens, but the lens is adjustable, right? It can become flatter or more curved, depending on whether you're looking at things that are close up or far away. And so light will bend a little bit more when we get to the lens. And from there, right, we're headed back towards the retina, right? And now the exact spot on the retina that you hit, that's all about focus, and we'll get to that later. But the idea is all of this intervening space in the middle that's just shown as being open, that's just a bunch of fluid, right? And that fluid is there in the so-called vitreous chamber. The fluid itself is sometimes called vitreous humor. And this is just a way to make the eyeball the right shape, right? The eye has to be roughly spherical, and so we need something to prop it open. That something is some fluid that has a little bit of pressure to it, right? The end goal is we want to get our light focused specifically on this layer. They're peeling it away here so you can see. But everything they're showing you here in yellow, that's the retina. That's where you find rods and cones. The red tissue behind it is choroid. That's just blood vessels. It's also what gives images red eye if it's not removed, because the red light reflects back off the blood vessels. Okay, but the photosensation, rods, cones, all of that business, that's happening in the retina. And then action potentials, right, signals from those rods and cones are going to go back axons that become the optic nerve and make their way to the brain. Okay, so the site where the optic nerve attaches, because it's the origin of the nerve, and the retina's kind of flipped a bit backwards. I'll make sense of that later. You can't have any rods and cones there, and so the attachment of the optic nerve we call the optic disc, and that's a person's blind spot, because if there are no rods and cones, you can't actually detect light. Okay, so let's talk a little bit more about the lens and focus. So in this picture, this is your lens, and they're sort of amplifying the size of it a little bit. It's not really that big, but what we're trying to get you to see is you have the lens held in place by these suspensory ligaments, and those suspensory ligaments are going to attach to a muscle, right? That muscle is sometimes called the ciliary muscle. They have it called retractor lentis on here. That's kind of an outdated term. That ciliary muscle can contract or relax to change the shape of the lens. Okay, and it's a bit difficult to picture, but the muscle is actually circular, and so if the muscle contracts, that actually makes the lens bulge and get flatter, because the muscles are ring, and so it becomes smaller. Lens gets more bulged and curvy, very similar to what they're showing here. That's for when you're looking at something very close up, because the light waves need to bend more to bend them back towards the spot on the retina that we want them to hit. If you're looking at something far away, right, distant vision, the lens is going to flatten, and the way that you do that is you relax that ciliary muscle. That would make the whole thing expand, and the lens would look more like this, right? And so the front surface of it becomes more flat, and that means the light doesn't bend as much. And so your lens really is very literally changing shape. Parasympathetic nerves are responsible for the contraction. That's when you're doing something up close, like reading or having a conversation, relatively peaceful, hopefully. Sympathetic input relaxes the ciliary muscle. Think like fight or flight, scanning the horizon, trying to look at things that are far away. All right, so now that we've got our light bent and headed in the right direction, we're going to zoom in and look very closely at what's going on in the retina. Right? This picture is an actual micrograph, right, from a microscope, and it's showing that this is the back of the eye, back here. This is the direction light's coming in from the front of the eye. And what's odd about this is the rods and cones are actually buried fairly deep in the retina. It's organized in a way that feels very backwards. Right? These bipolar and ganglion cells, they're the cells that are eventually going to carry the signal to the optic nerve and out to the brain. Right, so you can see it a little better in this illustration. These are your rods and cones, right, in this layer. So the rods that they're showing in green and the cones, because they really do have a bit of a cone shape, those are your photoreceptors. So what's backward about this is, again, the light is coming in this way. So your light wave has to get through these bipolar and ganglion cells that are not photoreceptors before it can hit rods and cones. Once you hit rods and cones, they can be stimulated by the light, and they're going to send action potentials. And so the end result of this is, once light hits a rod or cone, we expect to send action potentials, right, through the bipolar cell into the ganglion cell, and then the axons of those ganglion cells are what become the optic nerve. Right? That is actually going to take our signal back to the brain. And so the way that I'm saying this is backwards is that the ganglion cells are like the wires. It would make much more sense if we flipped this whole thing 180 degrees and made our wires, our ganglion cells, come off the back of the retina. Then you wouldn't have to have a blind spot. But this is the way the system evolved. This is kind of what we're stuck with. And in general, it works because this bipolar and ganglion cell layer is pretty thin. Okay, so we've got light in the retina. We're hitting rods and cones. We're activating ganglion cells. And now we just have to add a few more details. One of those details is that cones especially are not equally distributed throughout the entire retina. They're concentrated in a particular area. And that area we call the fovea centralis. So in this picture, what you're seeing is like the retina, like from the inside view. This somewhat larger area of high density cones is called the macula. And then this area of extremely high density cones right at the center is called the fovea or fovea centralis. So if you're out here, like these areas of the retina are where you'd be seeing your peripheral vision. They have a very low density of cones. They're mostly rods. The fovea and the macula, very, very cone dense. And so this graph is showing you 0 represents like right on the center of the fovea. At that point, right in the middle, your cone density is approaching like 175,000 cones per square millimeter. That's a dot, like, about that size, really, really tiny. And what this gives you is very, very high resolution, right? Just like pixel density in a camera gives you high resolution, cone density in the retina gives you high resolution. So when you're, for example, reading words on a page, the reason that your eyes track across the page is so that the words you're reading always hit the fovea. And if you try it, like, try focusing over here on the screen, like keep your attention right there. And then in your peripheral vision, see if you can read these words. It's not going to go very well because those words are hitting outside the fovea and you don't have as high a resolution, right? We call that idea tracking. The muscles of your eye are tracking words on a page, a person's face as they walk and you're following them, a football that gets thrown in a game that you're watching. Your eyes track the object that you're interested in so that you can see it with the highest resolution possible. Okay, so the next question to ask is, once light does hit rods or cones, how do they actually know the light is there, like from a detection standpoint? And the answer is your light sensor is this molecule called retinol. And retinol is a molecule that's derived from vitamin A. That's where all the eat your carrots or you'll go blind stuff comes from. Is if you're extremely vitamin A deficient, you can't make retinol because that's the substrate that it's built from. And rods and cones won't work. So the idea is this retinol actually exists in 2 different forms, cis-retinol and trans-retinol. And the key part of it is light is what makes the molecule change shape. Right? And so when light hits the retina, the first thing that happens is you change the structure of this molecule. It's not a chemical reaction. It's just a shape change. Right? The bend at this particular bond can either be straight, which would be the all-trans version, or it can be cis. Right? And the diagram is showing you is at rest, we've got the cis version, and this photon of light hits and converts to the all-trans version. Right? That is what is responsible for, signaling to the rods and cones that light is present. Right? Now, the confusing part about this is, and it feels incredibly backwards, but in the end of all this, having light present and converting retinol from cis to trans actually winds up hyperpolarizing these cells. Okay, so in a way, light actually turns off rods and cones, which again, I know feels incredibly backwards, but it'll all make sense in a minute. All right, so let's think about what that means in the actual retina. So if you add light, it really just means that retinol changes shape and your rods and cones are turned off. The reason that that makes sense, though, is this synapse between rods and cones and bipolar cells, this is an inhibitory synapse, right? It's inhibitory, which means when you turn off the rod and cone, you're effectively turning off inhibition, right? And that makes a double negative. It's like letting off the brakes in your car. You can go faster now. And so paradoxically, it seems like when lights turn on, rods and cones are off, but that just means that you stop inhibiting the bipolar cells. So the bipolar cells are actually turned on, right? And they have a normal excitatory synapse with the ganglion cell. Okay, so bipolar cells turn on, ganglion cells turn on. And remember, the ganglion cells' axons become the optic nerve, right? Cranial nerve 2. And so in the end, when the lights turn on, you get signals going to the brain, right? Your brain sees that there's light present. It's just through a double negative mechanism, right? Rods and cones are turned off, stop releasing inhibitory neurotransmitter. That means bipolar cells are turned on, ganglion cells are turned on, and your brain gets the information that it needs, right? On the flip side, if we turn the lights off, that would just mean that rods and cones would start releasing neurotransmitter onto the bipolar cell. But again, it's inhibitory. And so bipolar cells and ganglion cells will now be off, and the brain would not be getting action potentials down the optic nerve, right? Exactly like we would want if lights are off and nothing's happening. All right, so at this point, we have a signal going back to the optic nerve. We just have to figure out how it works. All right, so remember in the somatic nervous system, everything is contralateral, right? Left side of the brain controls muscles on the right side of the body, and vice versa. For the eye, it's contralateral, but it's not contralateral by each eyeball. So it's not that the left eye goes to the right side of the brain and the right eye goes to the left side of the brain. It's contralateral by visual field, right? And so what they're showing you here is any object that you see on the right side of your field of view, like objects that are over here, they all trace to the left side of the brain, right? That's why all this green area over here, if you trace the green color, that's just showing you the axons that they follow, right? And they wind up in the left visual cortex and the occipital lobe, right? Anything shown in purple, right? That is showing you things in your left visual field over here. Those purple axons trace to the right side of the brain. And so vision is contralateral, but it's contralateral by field of view, not by eye. And in order to make that happen, right, since each eyeball can see a little bit into the other field of view, that's why each individual eye has to have a fork in the optic nerve. And so when the optic nerve leaves the eye, it gets to this crossroads that we call the optic chasm. And that little fork, that crossover, is showing you that from each eye, about half of the nerves or axons cross the midline and go contralateral, and the other half stay on their side and go ipsilateral. But again, the retina is mapped out so that it's entirely dependent on which half of the field of view the rods and cones that are connected to those axons are viewing. Okay? And so for that reason, if you see something in your central field of view, it will very much go to both halves of the brain at the same time. It's only going to be one side if you're seeing something way out here in your peripheral vision that's only seen by one eye. All right, our next issue is telling colors apart from each other. So color discrimination just meaning identifying different objects based on what color they are. And just remember, color isn't really a real thing. Color is just wavelength of light. And so the wavelength of light in nanometers is just a physics property, right? So, ultraviolet light would be down here. That's even less than the wavelength we can see. And infrared would be out here. Like even radio waves are out further to the right, like X-rays are further down to the left. And so the light that we see isn't special. It's just electromagnetic radiation, right? Like there are some like insects, like honeybees can see UV light because they have a photoreceptor that can detect it. We can't. Some nocturnal animals can see further into the infrared spectrum, helps give them something that resembles night vision. We can't do that without technology. And so color is just how your brain sees wavelength, right? So if you're looking at an object that reflects light that has a wavelength of 400 nanometers, we call that purple. And if you see something with a wavelength of 600 nanometers, we call that orange, right? And in order to tell the difference, you have to have different types of cones, right? Blue cones just means these are cones that respond best to wavelengths of light that are shifted towards the blue end. And so their peak, like they're best at absorbing light. Absorption, think of as just being activated. Blue cones are activated best at around 420 nanometers. And then they taper off a little bit, but they still work out here, right? Green cones have a peak at 534, red cones at 564. And so when you see a particular object that has a color to it, All your brain is asking is, okay, which of these cones got activated, right? If you see something that has a wavelength of, let's say, 500 nanometers, that is going to give us minimal activation of blue cones, but it's something. Let's call that 15%. It's going to give you higher activation of red cones. Let's call that 55%. And even higher activation of green cones. Let's call that 80%. Okay, that combination, 15% blue, 55% red, 80% green, this 500 is the only wavelength that can get that exact combination, right? Because if I shift this way, blue activation will go up, red and green will go down. If I shift this way, the blue will go down and red and green will go up. And so no matter what wavelength of light you're seeing, it's going to activate a very specific combination or ratio of blue to green to red cone activation. And that's how your brain knows the color, is by comparing the three different types of cones against each other, right? People that are red-green colorblind they're either missing or have non-functional green cones. And if you don't have the green cone, you can still see these green objects because the red cone extends into the green area. You just can't tell the difference between green and yellow and red. People that have color blindness usually describe things in this area as just looking kind of gray. They can see it. You just can't tell the difference between different colors. All right, and last issue with vision. Now we're outside of the eye. We're talking about muscle movement. And your eyes moving to follow an object. Remember, that entire concept is called tracking. You should remember that from the macula and the fovea when we said we have to move the eye to make sure that the thing that we're seeing hits that sweet spot, the fovea on the retina. And you actually have 6 muscles around the eye. These are called the extrinsic eye muscles. I'm going to focus on 4 of them that are the most straightforward. All of these muscles that are called rectus. Rectus just means they connect to the eye at a right angle. And it's pretty straightforward. The superior rectus will pull your eye like backward so that you look up. The inferior rectus will pull the bottom of your eye back so that you look down. Medial rectus will make you look medially, and lateral will make you look laterally. Just remember, for up and down, it'll be the same in both eyes. So if you look up, the superior rectus of both eyes would be contracting since they both look up. If you look at something on your right side, well, the right eye would be using its lateral rectus because the right eye would be looking laterally. The left eye would be looking medially, so you would use the medial rectus. Right? If you contracted both medial rectus muscles, you would be trying to look at the tip of your nose, like both eyes in. And if you contracted both lateral rectuses, most people, I've never seen anyone be able to do it, but both of your eyes would be looking out, like very alien-esque eye movement where they're detached from each other, right? You also have a couple of oblique muscles, inferior and superior oblique. Those are more stabilizers because if they really moved, your eye would rotate like a radio dial. We're obviously not doing that. So these muscles are all controlled by cranial nerves. Remember, cranial nerves 3, 4, and 6, oculomotor, abducens, etc. Those are the cranial nerves innervating these muscles, and they control the eye movement to determine what you're looking at, which affects where the image hits the retina. All right. As always, hope this was helpful. Follow along in the playlist so you get the videos in order. Everything will make a lot more sense that way.